So let's try this again. I thought I would record a video for the study guide. I figured that would be easiest to do. Um, so let's look at the lab 4. So basically, if you weren't there for the water lab, you should go to the video on YouTube. There's a link on Canvas that'll show you most of these experiments. But you should basically understand the definitions of cohesion, adhesion, surface tension, surfactant, things like that. And again, kind of understanding what each of these demonstrations showed. I don't think I did all these on the video, but with this one, the glass bowl of pepper, understanding adding the surfactant prevented the pepper from sitting on the surface because there's no more surface tension. Things like water striders and things like that, they actually sit on the surface and don't break surface tension. They don't technically float, or there is a difference between floating and just not breaking the surface tension. The jelly jar, again, it was surface tension of the water that held the water in the inverted jar with the screen and the lid. If I would have added soap to that, the water would have flowed right through. I didn't do that because I would have been spending all day trying to rinse the soap out. You have to get those really clean. Again, the balloon and the stream of water from red solo cup. We induced static electricity in the balloon, and the static charge of the balloon pulled the stream of water off of directly vertical. So you could tell that there is a charge of some sort in that water. So that the water is, again, another demonstration the water is polar. It's attracted toward that statically charged balloon. Again, the capillary action, it was demonstrated by the glass apparatus with the different size diameters, vertical tubes, the water being pulled the highest to the smallest diameter tube. Again, capillary action is a combination of both adhesion, water sticking to glass, and cohesion, water sticking to other water. Remember, it's the adhesion that pulls the water up the tube. The cohesion keeps the column of water constant, keeps water behind the water at the top of the column. Likewise, the acrylic yarn, or the pouring the water down the yarn, showed both. When we saturated the yarn, that was adhesion, the water sticking to the fibers of the yarn, and then we showed cohesion by being able to pour water down the yarn, which the water being poured was cohesing to the water that was already adhesed to the fibers of the yarn. Again, polar versus non-polar. Just know that the polar and non-polar will not mix. Oils and waxes do not mix with water. That's why you need something like a surfactant, soap. Remember, soaps and surfactants are amphipathic, meaning they're polar at one end and non-polar at the other. So the non-polar end would surround your droplet of oil, and the polar end would be sticking out and face the water to allow that droplet of oil to be washed away. I have no questions on the hydrophobic sand. Again, that was just play, basically. So let's take a look at Lab 5, PowerPoint. Okay, why is this giving me issues? Okay, there we go. Hopefully you guys are seeing Lab 5, mitosis. So with Lab 5, understand the definition between mitosis and cytokinesis, again, just definition-wise. And remember, anything we saw pro-metaphase, just include that as part of prophase. So know the phases of mitosis: prophase, metaphase, anaphase, telophase. Again, understanding cytokinesis, whereas there's that overlap. And again, the formation of the cleavage furrow and the cell plate is an actual cytokinetic event, but we're using that as our visual indicator of when we're calling it telophase. The G1, S, and G2, remember, is all part of interphase, which is not part of mitosis, but is part of the cell cycle. And again, if you kind of picture this as a face of a clock, the cell spends just a small amount of time in mitosis, it spends the majority of its time in interphase between mitotic events. Again, like this slide, there may be an extra credit question or two about the different types of microtubules. Just know them, by definition. That's all I would ask: polar versus astral versus kinetochore. I might give you a definition. Just give me one of those three. So again, stages of mitosis, anything before the cells are lined up on the equator, we will call that prophase. So like you see here, prometaphase, we'll just call that the late prophase. Anything before the cells being lined up across the equator, we call prophase. But that moment in time when the cells are lined up, we'll call metaphase. Anything after that, we'll call anaphase, up until the point where we see a cleavage furrow or the cell plate start to form. So be able to recognize the cells in prophase, metaphase, anaphase, telophase, either from these photos that you see here, these animal cells, or these cells here that are photos from the actual slides that we have in class. So be able to recognize prophase versus metaphase versus anaphase and telophase. Likewise, in the plant, be able to recognize prophase and metaphase, either in these photos that you saw here from the text, or photos from the actual slides we have from the onion root tip. And again, the questions that you saw in the lab manual, what sorts of tissues would you expect to see? A lot of mitosis in either a plant or an animal. Again, these examples are perfect. This plant came from a root tip. All right, root tips are actively growing, and the animals slides came from a whitefish embryo. All right, embryos are undergoing lots of mitosis. Those are two examples there. Again, metaphase in plants is a little bit more sloppy than metaphase in animals, but you should still be able to recognize this as metaphase. Anaphase, you see those chromosomes starting to pull apart. And then telophase. Again, in the animal, we're looking for the cleavage furrow. In a plant, we look for the formation of the cell plate as our indicator of telophase. Again, I won't have any questions on this mitosis of renal cells. It's just an interesting video to watch if you can. I think I must have closed that one. So let's see if I can open up the diffusion osmosis. So this is what we need. All right. So hopefully you're looking at the simple diffusion right now. Again, just no definitions. Simple diffusion versus osmosis. Remember, diffusion is movement of particles from a high concentration to low concentration. The other way of stating that is down a concentration gradient. Remember, if you're going up a concentration gradient, you're putting in work, and that is not diffusion. Diffusion occurs spontaneously. There's no work to be put in. Now, some of the factors affecting the rates of diffusion, two of these we tested for in class. The temperature was the purple potassium permanganate solution at the front of the room. Remember, the heated solution diffused much more rapidly than the cold solution, and the steepness of the concentration gradient was also demonstrated by the dialysis bags. If you remember, the dialysis bags had 0.0 molar, which is just tap water inside the bag, tap water outside the bag, 0.2, 0.4, 0.6, 0.8, and 1.0 molar sugar solutions. So that is a steepness of concentration gradient. The most steep was the 1.0. The least steep, besides the one that had no concentration gradient, was the 0.2. Hence, the 1.0 molar solution at every stage gained more weight than all the other solutions. It was the steepest concentration gradient, hence had the highest rate of diffusion. Technically, we're looking at osmosis here, but we're looking at diffusion of water, really. But the same principles apply. So those two, steepness concentration gradient and the temperature, were two factors that we tested for. You should know these other factors just to know, but you should also know the ones we tested for in lab. Osmosis is just basically diffusion of water, but osmosis requires a selectively permeable membrane. Usually we talk about diffusion of a particle in either air or water. Osmosis really is just diffusion of water, but because water is just ubiquitous, there's a membrane that's got to be involved. And that membrane, selectively permeable, means that water can pass through, but the other, in our case, sugar, does not pass through the membrane. The other example of selectively permeable membrane could be the membrane of a red blood cell that we talked about quite a bit. You should understand why this is crucial for consideration of IV fluids. Again, we'll talk about Emily Cherry towards the end of this, but that could be an extra credit question there. So side B on this diagram represented what was inside of our dialysis bag. It was just water and a solute of some sort, in this case sugar. Side A represents what the solution outside of the dialysis bag, which was just plain tap water. So our dialysis bag was hypertonic compared to the solution around it. Or you could say our dialysis bag was in a hypotonic solution. You should remember those terms are comparative terms. And so it's not just, you can't just say this solution is hypertonic. Well, hypertonic to what? You've got to have X was hyper, isotonic compared to Y. So you've got to, it's a comparative term. You've got to have both things there. Osmotic pressure and reverse osmosis, you should understand these just straight by definition. Remember, osmotic pressure is the pressure that is applied by that dissolved solid to draw water across that semi-permeable membrane. And I remember we used reverse osmosis, applying mechanical pressure to reverse the osmotic pressure and to actually filter water across the semi-permeable membrane. This is how you turn salt water into fresh water. Don't worry about the osmolarity slide. I won't ask you about osmoles or osmolarity. Just understand that tonicity and osmolarity are kind of related. The more dissolved solids you have, the greater the tonicity or the ability of a solution to affect fluid volume and pressure of the cell. So if you drop a cell in a hypotonic solution, the solution around the cell has less dissolved solids in the cell. Water is going to infiltrate that cell, go into the cell, cause that cell to swell. Remember, water follows salt. Water follows the dissolved solid. So if there's more dissolved solid inside the cell than outside, water is going to rush into that cell, cause that cell to swell. A hypertonic solution, again, our example of that was dropping a red blood cell into a 23% hypertonic saline solution. That was, again, our example with Emily Cherry. So if you put a cell in a hypertonic solution, the cell loses water. So remember, water follows salt. So if there's more salt outside the cell, water's going to leave the cell, and the cell will shrink or crenate. What we want to aim for, again, is an isotonic solution, same amount of salt inside and outside. So our IV fluid that is isotonic is what we call 0.9 normal saline. And that's what Emily Cherry's medication should have been in instead of the hypertonic 23% saline. So again, looking at what your red blood cells look like, you should be able to recognize each of these. Again, a hypotonic red blood cell is swollen because it's been placed in a hypotonic solution like distilled water. Remember, the isotonic is 0.9, so the isotonic cell in the middle is in a 0.9% solution. So there is as much water entering as there is leaving. That's you're at an equilibrium. And the cell on the right has been placed in a hypertonic solution with a lot more dissolved solids than inside the cell. And that causes water to leave the cell, and the cell shrinks and crenates. And the spikiness that you get is left over from the cell cytoskeleton kind of exposed as the cell membrane shrinks. So if we go to Canvas, take a look at Canvas. what you have here under week 6, again, water chemistry. There's a link to the videos that I posted on YouTube. If you weren't there for those, the water experiments, you should kind of understand what we did and the significance of each of those experiments. Under Lab 5, again, the mitosis PDF, basically just understand the cell cycle, the phases of mitosis, and what both an animal and plant cell look like in each of those phases of mitosis. And with regards to week 8, again, understand diffusion, osmosis, and how you move water, hyper, hypo, and isotonic. Definitely want to understand those concepts. Again, I think I put a lecture on there for those who weren't in class, but that's what that link is there. Plasmolysis of Elodia, again, a link to the video we saw. So plasmolysis refers to putting a cell in a hypertonic solution. Again, this is kind of paralleling what we saw with Emily Jerry. They dropped her basically put a hypertonic solution into her blood vessels, and exposed her red blood cells to a hypertonic solution. So her cells lost fluid, just like you see in this plasmolysis of Elodia. They initially put 20% sucrose solution around these Elodia cells, and you see the cell membrane shrink and pull away from the cell wall. Then they replace that with a distilled water solution, which is hypotonic. And again, a hypotonic solution will cause the water to rush into the cell and swell. Remember, water follows salt. So whatever side of that membrane has the greater amount of dissolved solids, the water will move towards that side of the membrane. Water wants to dissolve. And again, a link to the Emily Jerry story. I will have an extra credit question regarding that. Maybe a simple, tell me in 1 or 2 sentences exactly, how it is that Emily Jerry died, and just explain what you know about a hypertonic IV solution and what that will do to your red blood cells. So hopefully that will prepare you guys for the practical 2 this week. Again, if you have questions, by all means, just email me, let me know.